Good morning, everyone. So we meet again after I think 3 or 4 weeks. Oh, 5 weeks I think, because I met you during at the week you did your midterm exam. I think it's about 5 weeks ago. So for the last 2 weeks of cell biology course, I will leave to you the information involving in the cell cycle. So as you know, in I think from the first lesson, that the only way to make a new cell is to duplicate a cell that already exists. So that is a statement in the cell theory, which was built in about 18th and 19th century. So when the scientists prove this, it carries with a profound message for the continuity of life. And all living thing, all living organisms, from the unicellular like bacteria to multicellular mammal like us, are products of repeated round of cell growth and division, extending back in the time to the beginning of life on Earth over 3 billion years ago. So for now, the cell continuing to do the division to maintain the stable of our life. So the cell reproduce by performing an orderly sequence of events in which it duplicates its contents, then divides into, and the cycle of duplication and division, we call it is the cell cycle. And for today, we will concentrate on the one very important part of the cell cycle is the cell division. So the cell division is one action of the cell for one cycle, and whenever they repeat the cycle, they also repeat the cell division. So somehow the time between the two division of one cell, they call the doubling time, or a time of one cycle of one type of cells. Um, for us, the multicellular organism, we develop from a single cell at the beginning, the fertilized egg, the most powerful stem cell in our body. Of course, now in the adults, this stem cell is not present, but we have the adult stem cell. But for the first cell, in the during the development, it is most powerful. They call the potent. They call the total pluripotent. No, total. Yes. Potential of the stem cell. And from this first cell, they continue to do the cell growth and division. And this action always happen at the appropriate times and in the appropriate places. And any mistake or any defect in the process of cell growth or division can lead to the abnormal cell, the cancer cell, or can trigger program cell death. In this image, this is illustrates the cell division at the early state, very early state, after the egg is fertilized to make the zygote. And the zygote continue to make a first cell division to make the two cell, and then the two cell cleavage to make the 4-8. And from the more than 8 cell, it calls the modular stage, or in Vietnamese it means the moral state also means the full cell. Okay, so in the biological development lesson, or in the biological development course, you will understand more about this stage during the early development of the body. And here I would like to show you the image of the cell division at the early stage of the development. And you can see the cell division, they can repeat and repeat many times. So on cell, it seems like synchronized to divide.
In an early Drosophila embryo, nuclei divide rapidly and in perfect synchrony. In this experiment, both DNA and tubulin are visualized with different fluorescent dyes. After the mitotic spindle has assembled, the microtubules, shown in green, start pulling the blue chromosomes to either pole. The chromosomes decondense and fill the newly formed round nuclei. In preparation for the next round of mitosis, the centrosomes duplicate and migrate to opposite poles of each nucleus, where they form new mitotic spindles, and the process repeats. The whole embryo rhythmically contracts with each division cycle.
So through this movie, you can see the very beautiful mitotic spindle is made from the microtubule and stay in the green color. And based on the behavior of the mitotic spindle, you can guess about the subspecies in the cell division. Um, but one thing I would remind you in this video clip is that you can see the time for cell division is quite quick, and the time between the two division is also very short, because during the early phase of the development, um, the cell will not spend much time in the cell growth, but it already concentrated on the cell division. So on the phase involving in the cell growth will be shortened, and they spend time for the cell division in order to make as much number of cells as possible during the early phase
In
of
an
the
early Drosophila
development. So cell mass composition during they been copying the cellular components and then dividing the cell so that the components are distributed evenly to the daughter cell. So the action of copying cellular components, they involving in the DNA replications and organelles, increase the number of organelles or structure or the other basic molecule like protein, lipids, or glucose molecules inside the cell. So the alternating of the growth and division activities of a cell is called the cell cycle. um, I think you already knew that one cell cycle contains four main phases. The name is D1 phase, S phase, and M phase. And among of them, the growth activity corresponds to the interphase, which conclude the G1, S, and G2 phase. Also, one thing I want to notice you is that the cell come to one very specific state. But in this state, so this cell is somehow like the cell will not divide, and they call the dividing cell. And the state that the cell come and stay and not divide, they call the Z0 phase. So Z0 phase somehow they call the quiescent state of the cell, or the quiescent phase of the cell. Um, But it not mean that the cell do nothing when they enter the Z0 phase. In fact, they place function. They place their function when they stay in the Z0. And most of the differentiated cells stay in our body stay in the Z0. So it's like the skin cell, whenever they come to they will stay in the Z0 phase, and then they do the protection function, or the cell made up the gland in our body, they will secrete the hormone or the other molecule during their function, and they are in the Z0 phase. Our neuron cell also stay in the zero phase, and muscle cell also stay in the zero phase. On all of them, they do function every second, every time, and they stay in the zero phase. So, do you know the percentage of the dividing cell? Can you compare the percentage of dividing cell, or the number of dividing cell, and the number of non-dividing cell in our body? Which is um, which is more? Which number is higher? The diving cell or the non-diving cell? Dividing cell or non-dividing cell? Sorry. Which number is higher?
The non-dividing cell, I think.
Hmm. Okay. Any ideas? Yeah, obviously you can see when I give example, you can see that most of our cells in the body, they belong to the non-dividing cell, and only some of them, very a small group of the cell in each type of cell can every hours the cell in our body can be die by apoptosis. For example, the cell in the intestinal epithelial layer in the intestinal system, the cell can die every 12 or 20 hours. So the stem cell for this cell, they have to divide and make the new cell. Our skin, I'm sure you understand that our skin, they lose the cell as the this on the surface of the skin, right? Even the first layer is not exactly the cell, but the keratin layer, but also the cell from the deeper layer, they will differentiated into the keratin layer, and whenever the keratin layer remove, and the other layer, the sublayer, they continue to development and differentiate into the keratin layer also. So they must have another cell to continue to divide and to provide the new cell and maintain the differentiation and maintain the thickness of the skin. And based on that, they can maintain the function of the skin as a protection barrier in our body. So the cell lose and the new cell is appear, and this for cell division cell or cell progenitor or progenitor cell. So which activity the cell will do in each phase of the cell cycle? This already give to you when you was at high school, I think in the class 10, I think. So I will briefly introduce you about the main action happen in each phase of the cell cycle. So for the Z1 phase, One thing I want to tell is about the name of the each phase. So the M phase come from the mitosis, and the S that they come from the synthesis. So the name also, how to say, indicated the main action during this phase. And what the Z stand for, the Z letter stand for. So it's interesting because the Z stand for the gap. So why the scientists call it two gaps? Because at the beginning they just see the cell, they just observe the cell division and then the cell replication their DNA, and they don't know exactly which thing they shall do in between the M phase and the S phase. So at the beginning they name that the two gaps, gap 1 and gap 2. But now we all knew about the action of the Z1 phase. So as I say, this in the during the Z1 phase, the most important work is cell growth. So the cell they just come from the end of the M phase and they quite small, and the daughter cell need time to grow and increase in their size, also in the number of organelles and the other structure or molecule inside the cell. It's somehow like the baby was born, they need time to grow up and become the teenage, and then the adults. So they need about 18 years to do that. For the cell, also the one phase is quite the longest phase during the cell cycle. And if the cell not if the cell come into the Z0 phase, as I already said before, so the cell will lose their function when they stay in Z0 phase. But if the cell continue to their cycle, so they will prepare the chromosome for the replication which happen in the S phase, and also they will duplication the cellular component this phase. And in the Z phase, this from the cell cycle to come into the Z0 phase. And if the cell successful to pass from the Z1 phase to window in the M phase, so centrosome is duplicated in the S phase. Um, so that is the two main important action in the S phase. And for every cell division or every cell cycle, of cell growth is the DNA replication. So sometimes the but they have to duplicate the DNA before they do the cell division. So that is the most and for the Z2 phase, in the DNA or chromosome because they already finished the cell, the DNA replication. So now the number of DNA is double, and also the two centrosomes have formed in this phase. Um, continue to be synthesized, and some organelle also can be in the Z2 phase is in the Z2 phase is spent for the checking. That means it will check if all the DNA is already replicated, and is there any defect or mistake during the DNA replication? So whenever they find out something happened, they can stop the cell cycle and repair the mistake, and only that the cell can continue to the next phase. And for the next phase, it is the M phase. So the cell division happen in the M phase, and the division, the ability of bed distinguish the living thing from the non-living matter. And the continuity of life is based on the reproduction of cell or cell division. And the cell division is integral part of your knowledge. So what is the subspaces in the M phase? Do you remember? So I will give the question to all of you. In the classroom, do you understand the subphases? I see only four of you give the answer, and now it's increase in nine. Nine one. Okay, so let's have a look. Quick look. Okay, so none of you have the complete answer, but I think if I take all of the answer, it is complete. Why I say that? Because like one say Qi, right? Or Ling Qi, she say there are two subspace, the mitosis and cytokinesis. Yes, because the mitosis is nuclear division and cytokinesis is a cytoplasmic division. Yes, you're right. But also you should indicate it in more subphases in the mitosis. That means the nuclear division, and the subphases in the mitosis is like tuệ anh is a prophase, prometaphase, metaphase, anaphase, and telophase. Okay, so that is the subspaces in the M phase. Okay, so the M phase has the two. In fact, for now, they divide the M phase into two stages. Two stages. One is mitosis, and the other is cytokinesis. Somehow is equivalent to the telophase. Yeah, it's dry in most of the case of the cell type, but in some cell types or in some state of the cell division or the development of the body, the cell will not complete the cytokinesis. Even they do the telophase, but they not divide the cytoplasms. They stop dividing the cytoplasms and wait until they receive the signal from outside, and then the cytokinesis start. So because of that, now the scientists divide the M phase in two stages, the mitosis and cytokinesis. And for the mitosis, they can compose four subphase, five subphases, five subphases, as I just tell you. And the telophase, for example, in this slide, you can see here the cell, the change in the cell morphologies, and the structure is formation. The new structure is formed during the cell division. So, for example, here it is interface somehow like the Z2 phase, because you can see the two centrosome, and then the prophase, prometaphase, when the cell membrane start to disappear, metaphase, the early anaphase, late anaphase, telophase, and cytokinesis. So this cytokinesis and telophase in most of cell type, they happen together at the same time. So somehow it's like the cytokinesis is one part of the mitosis. But in fact, in the other cell type, they can separate it into stages, and it is called the cytokinesis. Okay, so I have another question. It's very popular question for the cell cycle. Anytime you work with the cell cycle, you have to perform this kind of experiment. So I will also leave the question in the classroom. So like this, you can answer in the classroom. In the question of the classroom, I did not leave the image, so you can look at the image in the slide and together with the question to give the answer. Okay, so the question is, if you have a cell culture, that means the cell isolated from the body, and then you can grow the cell in the laboratory in some culture dish. I'm sorry. So you want to know how many cells stay in each phases of the cell cycle, right? So you have to perform one technique they call the flow cytometry. I'm sorry, wait me for a second. Okay, so you will take all of the cell from the culture and you stain the cell with a fluorescent dye. For example, like DAPI or H, the name of the DNA fluorescent dye. And this dye, this fluorescent dyes will bind to the DNA. So the the more number of the DNA, the more intensity of the fluorescent. Okay. After staining the cell, so you will apply the cell onto a flow cytometry system, or they call the flow cytometer. And this cytometer, they can measure the signal, the DNA, the fluorescent signal. And because the fluorescent signal correspond to the DNA, so somehow they can reflect the number of the DNA in the cell. So after you apply on the cell on the flow cytometer, so the number of cells with a given DNA content is plotted on the graph as you see here. So this graph, it illustrates the DNA number or DNA content in each cell. So on the dot plot in the A, in this part it belongs to the A area. We name the A area. So all of these parts, they have the similar or they have the same DNA content. And similar in the B part, so this in this part, all the cells have the same DNA content. Okay, so based on this graph, can you indicate which phase of the cell stay in? So in this histogram, the vertical is the number of cells and the horizontal axis is a relative amount of the DNA per cell. Okay, I see already 11 students give the answer. And the answer is very, how to say, well distribution in the three choices. A two five four, A 1 2 0 bits. Okay, okay. Interesting. Okay, so one thing I would remind you here is that when the cell stay in the M phase, and even they do the telophase, they stay in the telophase or cytokinesis. So whenever the cell still stay in the cytokinesis, not finished yet, the two daughter cells still bind or still link to each other through the structure they call the mid-body. So when you apply the cell on the cytometer will consider or clarify this is one cell. This is one cell, okay? Because they still link to each other, they're not separated yet. So whenever they separated successful, so they come to the Z1 phase. But still they linked to each other, they still belong to the M phase. Okay, so that is the thing. We have now 15 answer. 15 students already turned in. So what is the right or the right answer? So the right answer is the second choice. That means the pick A represent for the Z1 and Z2. The pick B represent for the Z2 and M phase, and the between pick A and pick B is the S phase. So why? Because in the pick A, it is the last or the least DNA content in per cell. And the Z2, the B represent for the cell which have more DNA or the highest DNA content per cell. So as I say in the what the cell do in each phase, so the cell have the, for example, like human cell, they have two ends of the DNA in the Z1 phase. But whenever they come and whenever they finish their DNA duplication, so they will come to 4N, right? And this 4N will remain from the Z2 to the end of the M phase. So that is why the highest number of the DNA will stay in the Z2 and M phase, and of course they will be in the B peak, and the lowest is in the Z1 or Z0 peak. For the S phase, during the S phase, the cell will start to replicate the DNA. So each DNA molecule will start to replicate at maybe different time point. So that is why during the S phase, you cannot get the peak because the number of the cell with the same DNA content is quite equal. It's quite equal between cell. So you have one you have the phenomenon of the S phase is not the peak. It's quite flat because the number of cells at different DNA content is quite equal to each other. So you see that cannot have a peak here. So based on the DNA content, you can figure out the number or the percentage of the cell in each phase in the cell population. And also because the high number of the cell of the Z1, Z0 phase, so this phase is the highest or the longest phase in the cell cycle. Okay, so that is the answer for this question. And whenever you work with the cell and you perform this cell cycle experiment, so you can know how normal of your cell population. Because in some state, the cell can change the percentage of the phase. For example, if you counter the cell and in the counter flash and you forget to take care of them, and the cell reach they got the highest confluence, that means they stay in everywhere in the flask and there is no space between the cell, so the cell will not divide. And when you take this cell to do the cell cycle, you will see that they always stay, or most of them, they stay in the A peak or Z1, Z0. And very, very few, or I can say no cells stay in the B peak. That means the Z2 or M phase. So this cell, somehow they stay in the Z0 phase, or they come to the Kirschen phase. They will not continue to the next cell cycle because there is no space surrounding them. So this is one signal to inhibit the cell division. Okay, so based on that, based on this technique, you can know that your cell population is in the normal state or in the stress state or in the abnormal state. And especially when you work with the cancer cell, you will use some drug to treat your cancer cell, and you can see that whenever you add the drug on the cancer cell, so the cell cycle population changes. For example, when you add the paclitaxel or paclitaxel on the cell site on the cell cancer cell, so the cell cycle will not look like this. They change a lot. I don't know if Dr. Long already introduced to you about the paclitaxel, onocorazone, or colchicine, but all of these drugs they affect the cell cycle. So okay, the M-phase can divide into stages like mitosis and cytokinesis, and the mitosis is divided into 5 subphases. That they are prophase, prometaphase, metaphase, anaphase, and telophase. And we check the behavior of the cell in each subphase, but mostly I would like to concentrate in the two structure of the cell division. Here is just the image shows you the main action of the cell during the subphases of the M phase of the mitosis. In the upper image, it is a real cells stained with the fluorescent dye. So when you look at this photo or these images, can you recognize what the green colors represent for which thing? Which thing the green color represent?
The aster
Yeah, the aster of what?
of the centrosome.
So that means the centrosome, right? And centrosome then could play function like the aster or the polar of the mitotic spindle. Yeah. So if you just say it's centrosome, it's not quite correct because here you see some filament, right? Also in the green color. It's not only the bright there, the very bright dot here, but also there are many filaments. So it connects molecule that the green colors represent. Okay, that is microtubule. Okay, so centrosome is one NTOC. Does that mean the microtubule MTOC? Okay, microtubule organization center. If you remember in the cytoskeleton part, so from the centrosome, many microtubule is formed, and that is why you see the very bright green as this part. That means the located of the localization of the centrosome, and from this centrosome, many microtubule grow to everywhere here. So you see the green filament. And what about the blue color? The blue color represents for which molecule? The blue one.
The chromosome.
Yes, the chromosome or the DNA. Okay, so when you look at the first photo in the left, this is a Z2 phase cell. That cell stay in the interface, but they stay in the Z2 phase. The centrosome is already duplicated, and at this moment they still stay together. And you can see, as I say, many microtubule growing from this centrosome. On those nuclear, you can see the very well defined nuclear with the contact membrane, and you cannot see the chromosome in this phase, but you just see the color is some like diffuse everywhere in the nuclear, because at this state the chromosome still stay as a chromatin fiber, and also they distribute everywhere in the nucleus. So you see the color, the blue signal is quite a well distribution in the cell nucleus. But when the cell comes to the prophase, when they enter the prophase, so first the centrosome start to separate, and now you see they are not as bright as in the Z2 phase, but they seem like enlarged of the green signal at this part. It is because the centrosomes start to separate from each other. And also in the nucleus, you can see that now the color is not well distributed. You see in somewhere, some places they are more blue than the other part, because at this stage the chromatin start to condense. So whenever they condense, the fluorescent signal will also condense too. So they will concentrate in the DNA, not the other space. So you can see here the presence of some chromatin or chromosome, but not very well separate from each other. And when the cell come to the next phase, the prometaphase, you see there is no nuclear membrane surrounding the chromosome. The nuclear membrane is broken at this phase, and the chromosome is more visual. That means you can recognize the chromosome easier because they more condensed, and each chromosome seems more separate from each other. Also during the prometaphase, you see the two centrosomes now, they stay in the opposite side through the chromosome. So they will make the two polar of the mitotic spindle. And during the prometaphase, the microtubule will develop from the two polar to attach together and also attach to the kinetochore surrounding the centromere of the chromosome. So in the in the lower image, in the below image, you can see more clearly the behavior of the chromosome and the mitotic spindle. So in this state, during the prophase, the microtubule from the centrosome can come and bind to the kinetochore of the chromosome. And in the metaphase, now the chromosome successfully arranged as one line as a metaphasic plate. Two sister chromatid, and each chromatid will bind to the microtubule from one polar, and the other chromatid will bind to the microtubule from the opposite polar of the mitotic spindle. So on the chromosome now is highly condensed, or the most condensed, and they bind to the microtubule and ready to separate. So whenever the chromosome is ready, the anaphase happens and the two sister chromatids will be separated from each other to the two opposite spindle pole. So there are two equal groups of the chromosome separated apart from each other to the two opposite sides of the cell, of the mother cell. And then from the metaphasic plate, the cleavage furrow appear to separate the cytokinesis of the mother cell. So that it happens in the telophase and also in the cytokinesis. The cleavage furrow is formed also the nuclear the nucleus is formed in this way, and the nuclear membrane is reorganized surrounding the chromosome during the telophase. So that is the main action happen on the in the M phase subspaces, and I just briefly introduce to you about this. And the next in the next slide, we will concentrate to the formation of the mitotic spindle and how the mitotic spindle support for the chromatic for the sister chromatic separation in two equal parts during the anaphase and telophase, and how the cleavage furrow is formed to separate the cytoplasms of the mother cell. We will concentrate on these two structures in the M phase in the next slides. And now it's time for a short break. Okay, so we will back at 10 a.m.